Hi, everyone. Thank you so much for coming today. My name is Betsy Boughton. I'm the program director of agroecology at Buck Island Ranch. Before we get started, I just wanted to see if there was any new people we could introduce. I know I have one. Oh, Viv, you go first. Awesome. We have a volunteer at the ranch this week. Her name's Anna Wozni from UCF, and she's a senior at UCF and just is exploring different options for future um, activities, maybe an internship at Archbold. But thank you, Anna, for your work here this week. Um, Jenny Schaefer's in the audience with us. She's the old dog, but we'll produce her. Okay. So thank you everyone online for joining us as well. Today I have the pleasure of introducing Katie Niermann. And Katie has been an intern with the agroecology program for about 6 months now. Yes, so she's been a really great addition to our lab, always happy, cheerful, really nice to have that personality in the lab. Really hard worker, asks a lot of good questions, and we've just really enjoyed having Katie with us for her internship. So before Katie came to Buck Island Ranch, she graduated from University of Illinois Urbana-Champaign, and that was in August 2023. And there she did a senior thesis on solar development and land use, and she worked a lot with GIS and also did projects on environmental justice. So really diverse background. Today, she's going to talk about the impacts of diverse plant species on leachate volume and nutrient concentrations, and it's part of a larger project that we've been doing at the ranch. I also want to give a shout out to our lab for helping to mentor Katie, Karen Rice-David, especially Jackie Amartia. So it takes a village to mentor our interns, and I'm just really thankful for that. So now I'll turn it over to Katie.
All right, so hi everyone. I just wanted to thank you guys all so much for taking the time to be here and showing interest in my project. It really means a lot. So the title of my seminar is Impacts of Diverse Plant Species on Leachate Volume and Nutrient Concentrations, which is more of an applied soil health and water quality experiment. Oh yeah. All right, can you still hear me? All right, cool. So what exactly is leachate? Leachate is the downward movement of water-soluble nutrients across the soil horizon. Many of these nutrients happen to be bioavailable, which means that plants can access these nutrients to help them carry out their different functions and metabolic processes. Leachate is typically caused by an excess of water either through rainfall or irrigation. And leachate often flows into the surrounding ecosystem and can find its way into things like groundwater, rivers, streams, lakes, and coastal areas. And on the screen, you could see one of our leachate buckets from our experiment. So why exactly is leachate such a large issue in South Central Florida? A lot of this has to do with the region's soil characteristics, which are dominantly, we have dominant soil types such as spottizoles and alphazoles, and these tend to be pretty sandy, acidic, infertile, and have low surface soil phosphorus retention, which just further increases leaching potential. The high amounts of precipitation we receive are another cause. We receive approximately 54 inches annually. So this means our soils are typically seasonally inundated, which just amplifies this issue even more. So you can actually see with the picture on the top right portion of the screen what a typical spottizole soil matrix looks like. You could actually see this dark horizontal line right here, which is the spotic horizon. And this is typically where nutrients accumulate within the soil. So getting into some of the management practices contributing to nutrient pollution in water, there are many different contributors, for example, urban and cropland land use, but the one we're going to focus on and which is very prevalent in our area is ranching and agriculture. The management practices associated with this sort of land use has led there to be excessive ditching and heavy application of fertilizers. Nitrogen is a common fertilizer that is currently being used in a lot of these areas, but historically phosphorus has also been used. And so our area actually has a big issue with something called legacy phosphorus, which are characterized as deep accumulated stores of phosphorus within the soil. This has resulted from the long-term application of these phosphorus fertilizers, and actually the use of phosphorus fertilizers as a management practice has greatly reduced since the 80s. Yet even 40 years later, these deep stores continue to cause an issue when it comes to water quality. So why exactly is this important to us? Well, Lake Okeechobee is located downstream from us, so water from this region, which is primarily dominated by agricultural land use, flows directly into the lake, which then flows directly towards the Atlantic and Gulf Coast, along with the Florida Everglades. And in this picture on this slide, you could actually see this region's current water flow being visualized. So nutrient pollution from our region can cause major watershed issues downstream. This means that any excess of water-soluble nutrients that can find their way into the surrounding ecosystem can be a major threat to our vulnerable water resources. This could cause things like eutrophication, which is where we get things like algal blooms and dead or hypoxic zones, which can lead to things like organism deaths, such as fish kills, which you could see in this picture right here. And this actually isn't a new issue. Lake Okeechobee has persistently been experiencing noticeable cultural eutrophication since the 70s, which you can see in the top right picture right here. Lake Okeechobee actually undergoing a eutrophication event. I believe this is in like 2019 or 2021, one of those. So this could also pose a large threat to human health as water quality is directly impacted. It can impact things like our drinking water and some of the ecosystem services that we rely on. So overseeding is planting seeds directly into existing pasture land over the existing plants and grasses that are already there. This could help increase plant community diversity, which can also improve nutrient uptake. The thought behind overseeding with diverse plant mixtures are that this might help improve soil and water quality over time. So there's actually a project focusing directly on this that is currently ongoing at the ranch. It is managed by Karen Rice David in partnership with the Nature Conservancy, and it is also funded by the Florida Department of Agriculture and the National Cattlemen's Beef Association, so that this project is part of that larger ongoing project. So now getting into the research questions and hypotheses for this study. Our first question involves investigating how planting treatment impacts leachate volume. For this question, we are first hypothesizing that bare ground soil will produce the greatest volume of leachate. This is because we're predicting that lack of plant structures within this treatment won't be as good as preventing water from percolating through our pots and into the leachate bucket. So bare ground volumes will be higher than other treatments. We are also hypothesizing that between treatments, there'll be variability in terms of the volume of leachate that's produced. This is because we predict that species differences will impact a plant's ability to leach, as the difference in root structures could help capture water and nutrients in different ways. So for our second question, we'll be investigating whether leachate volume impacts nutrient concentrations. For this question, we are hypothesizing that nutrient concentrations will relate to volume either positively or negatively. Not entirely sure which case will be true, but we predict whatever the case ends up being will be due to there being either an accumulation or dilution effect on the nutrients, which can be associated with increased leachate volumes. And lastly, for our third question, we are investigating how planting treatment impacts nutrient concentrations. For this question, we are first hypothesizing that bare ground soil will have decreasing nutrient concentrations within leachate over time. And we're predicting this since there are no plants present that can either soak up any water or nutrients through the roots, and there aren't any above ground structures acting as buffer or slowing down our watering treatment. We are also hypothesizing that between treatments, there will be variability in nutrient concentrations found within leachate. We predict this because we assume that the diverse species belonging to different functional groups will interact with and process nutrients in varying ways. So why exactly are we asking these questions? Why do we want to know the volume of leachate and want to quantify the nutrient concentrations that are being leached? Well, these measurements are important when helping us estimate the nutrient loads entering our surrounding ecosystem and groundwater. Knowing these things can help us predict pollution potential, which is important to finding out, especially within our ranching and agricultural centered areas, as this can help us better guide management practices with the hopes of improved water quality. So getting more into the project specifics, the study site for this project is at the Shade House located on Buck Island Ranch. Buck Island Ranch itself is a 10,500 acre working cattle ranch, which also holds our agroecology research program and lab. We have approximately 3,000 head of cattle, and the land mainly consists of both semi-native grasslands and improved pasture habitat. So why exactly do we use the Shade House in this study? Well, it provides a controlled yet representative testing environment. It prevents larger animals from getting in and disturbing our treatments. It also has irrigation and sprinkler systems, which help us control the watering treatment. It also allows natural light and weather our atmospheric effects from the course, just like how it naturally would in the field. So the planted species we analyzed in the study include crimson clover, oat, daikon radish, hairy vetch, and bahia grass. We chose these species because of the different plant characteristics they exhibit. So we assumed they have functional diversity, and so we wanted to see how that would impact leachate volume and nutrient concentrations. These species are also being used in the overseeding study and have also been implemented into Buck Island Ranch pastures, so it just made sense to use them within the study. So clover and vetch are legume species, which you can see on the screen. Legumes have bacteria within their lower root nodules called rhizobias, which allow them to fix nitrogen, and so legumes tend to flourish within nitrogen deficient soils. Then we have daikon radish as our taproot species. Taproots can be pretty efficient at storing water and nutrients from the soil. And finally, oat and bahia are our annual grass species that we have. They're relatively efficient at accessing water and nutrients from the shallow portions of the soil due to their fibrous root systems. And also just a notable mention, bahia is the dominant forage species that we have on the ranch. It has actually been used in previous studies to help combat erosion and improve soil health. So we thought it would be a good species to add within this study. So this diagram on the slide shows our experimental design. In this experiment, we compared 5 seedling treatments right here, which were a winter seed mix and clover, oat, radish, and vetch monocultures, along with our 2 control treatments, which were bahia and bare ground. So each of these treatments had 3 replicates each, which was 21 pots in total. Our monoculture treatment had 8 seedlings per pot. Our winter seed mix treatment had 16 seedlings per pot, 4 seedlings of each species. Our bahia control had 4 sprigs per pot. Our bare ground treatment was just ground soil. So to test these treatments, we set up a nested pot mesocosm design within the shade house, which you could see this picture right here. Is our winter seed mix and pot positioning. So we used 2 2-gallon buckets. This bottom bucket acted as leachate reservoir, and this top bucket was fit with our soil, our treatments, as well as a soil liner to prevent any large soil particulates from falling through and contaminating our reservoir. We also taped the gap between the 2 pots, which you can see right here, to keep any little critters or water molecules out of our leachate reservoir. And so the soil we used for this experiment was also taken directly from ranch pastures. We sampled weekly over a 9-week period, and our pots were watered using overhead sprinklers that ran on a timer. Water treatment very dependent on ambient heat and rainfall. Generally, we watered 5 to 10 minutes per day, 3 to 7 days per week. Also, the pots were randomly rotated weekly, and the sprinklers were checked and cleaned intermittently to prevent any buildup, which is ensured that each treatment received an equal watering treatment. So the data points we collected and decided to use for the study include treatment, leachate volume, and nutrient concentrations. However, we did collect other data points such as above and below ground biomass, specific root length, volumetric water content, and temperature, which we could further investigate in the future. Nutrients we sampled for were orthophosphate, total phosphorus, total nitrogen, nitrate, and ammonium. Orthophosphate and nitrate tell us water-soluble, bioavailable concentrations of phosphorus and nitrogen. Forms of these nutrients are what can contribute to things like eutrophication. For total phosphorus and total nitrogen, this tells us all forms of phosphorus and nitrogen. both bioavailable and not bioavailable forms. And this is still important to measure since these other forms of phosphorus and nitrogen have the ability to convert to an organic bioavailable form over time as they naturally break down. And lastly, we have our NH4 or ammonium concentrations. This tells us the amount of ammonium within the system. This is important to measure as ammonium quickly undergoes nitrification in a natural system, which basically transforms the ammonium to nitrite, which then transforms into nitrate, which can also lead to eutrophication. So the weekly collection process consisted of 3 main phases. First phase was the preparation phase, which generally included getting all the equipment together that we would need for sampling. Second phase was the data point collection phase. So to start off, we would typically remove the tape from around the pots, and then we would shimmy our 2 nested pots apart. Then we would record the leachate volume, which you can see us doing here. And then we would rinse in between each sample with distilled water to help combat cross-contamination. We would also take a preserved and a filtered grab sample, which you could see us filtering right here and taking the preserved sample right here. And lastly, we would place all the samples on ice to keep them fresh and from any nutrients further breaking down. And after everything was complete, we would retape the pots for the next collection. So the third phase of this process was our post-collection process, where we package and sent our samples out to get tested. Then we would acid wash all of our equipment to ensure it was sterile for the next testing period. So we've performed all of our analyses in R, as you can see from the flowchart on the screen. This was our general data analysis process. We would start by testing for normal distribution. We soon discovered that none of our data was normal, so we tried to long transform and retest everything to see if there was normal distribution. This still was not normal, so we ran a Kruskal-Wallis test. And if the test found anything significant, we ran a post hoc test, which tested what factors were significantly different from them. So for the first question, we'll be analyzing how plant treatment impacts leachate volumes. So we'd hypothesized that bare ground would produce the greatest leachate volume, and that between treatments, there'd be variability in leachate volumes produced. When plotting the volume versus treatment, which we do right here, we plotted the volume on the y-axis, treatment on the x. And so we ran a Kruskal-Wallis test after doing this. We found that overall treatment was not statistically significant when it comes to predicting leachate volume. And this could be because the plants that we used were seedlings, so they're relatively small. So maybe the plant's roots aren't absorbing as much water compared to the rate in which water was coming in. But maybe if they're bigger, this would be a different story. We also do see bare ground having the second highest volume, which you could see right here. However, the volume it produces is relatively similar to the rest of the treatment volumes. We also saw a trend towards the radish treatment producing the greatest volume of leachate. This is interesting because taproots are supposed to be relatively good at storing water nutrients from within the soil. So what could be causing this is that there must be something about radish structure or function, potentially a taproot causing some sort of maybe like a pocket or channel within the soil that helps the water flow directly through. And so with this information, we can reject both hypotheses for question 1, which means that if we plant any of these other species, they aren't going to help increase or decrease leachate volumes better than any of the other treatments. So the second question we'll be answering is how leachate volume impacts nutrient concentrations. So we ran a Kruskal-Wallis test to see if there were any significant correlations between leachate volume and nutrient concentrations. So there was only an indication of significance within leachate volume for total nitrogen and ammonium, which is TKN and the NH4. We found that as leachate volumes increase, total nitrogen, TKN, concentrations significantly decrease. We think this could be because ammonium is converted to nitrate, so it is possible that this process is slowed down with greater leachate volume as oxygen is required for nitrification. So the greater leachate volumes, maybe this means that conditions are more anaerobic, and therefore nitrification would slow down. We also found that as leachate volume increases, ammonium concentrations significantly increase. So with this, we think that even if ammonium is increasing, maybe the other forms of nitrogen are decreasing within TKN, but however, this needs to be researched further. So we originally hypothesized that nutrient concentrations would relate to volume either positively or negatively. We found that this was true and significant in the cases of total nitrogen and ammonia. So lastly, the third question we'll be answering is how planting treatments may impact nutrient concentrations. We hypothesized that bare ground treatments will have decreasing nutrient concentrations within leachate over time. However, we found that only nitrate ended up decreasing over time. The rest of the concentrations for bare ground were highly variable every week. And graphs we have up on the slide are showing the average orthophosphate and total phosphorus concentrations in the leachate produced by each treatment. And we had the nutrient concentrations on the y-axis, planted treatment on the x. So we ran Kruskal-Wallis tests and found that our hypothesis was correct. We did see variability between treatments. There was variability in ortho P and total P. We saw that our mix contained lower concentrations of Bahia grass, yet we also saw that there was no difference between our mix along with the oat, radish, and clover monocultures. So overall, our mixed treatment had the lowest average concentrations for both total phosphorus and orthophosphate, and we're assuming that this must have something to do with the diverse plant species working together to prevent higher phosphorus leaching. So also for both total phosphorus and orthophosphate, we saw highest concentrations within both vetch and Bahia grass. We hypothesized that we were thinking that Bahia would have the highest P concentrations, though, as there's a previous study done at the Shade House that suggested Bahia produces high concentrations of phosphorus leachate. We also assume that there is something about vetch that is different from the other legume, as vetch is noticeably higher. You could see right here than clover, which is in red right next to it. So in this graph on the slide, this is visualizing the average total nitrogen concentrations found within leachate produced by each of our planting treatments. Once again, we see the concentrations on the Y and the planting treatments on the x-axis. We also found that our diverse species mix had high total nitrogen concentrations despite having low total phosphorus and orthophosphate concentrations, which is right here. The bare ground Bahia, radish, and mixed treatment So on the left, we see the phosphorus nutrient concentrations. On the right, you see our TKN concentrations. Once again, you can see that for phosphorus, the mix is has the lowest concentrations, but for nitrogen, it had. So we believe that planting a diverse seed mixture could be viable for improving water quality within this region, since we saw that seed mix treatments had the greatest impact at reducing phosphorus concentration. So studies actually suggest that controlling phosphorus nutrient is more effective at mitigating lake eutrophication than any of the other nutrients that we did that testing for. The Lake Okeechobee downstream, this is relevant to us. But even though it has very low concentrations of P, we also discovered that our mixed treatments had higher amounts of nitrogen than its leachate. So this points to a need for further investigation, where we could hopefully determine whether the benefits from having reduced P loads outweigh any negatives associated with increased nitrogen loads when implementing these species mixes. And since we also saw clover and oat having both reduced P and N concentrations, looking further into these species could be beneficial when the desire is reducing nutrient load to improve water quality. We also believe the usefulness of overseeding for improving water quality depends on successful establishment in the field. Anecdotally, we see higher establishment of mixed species in bare ground rather than compared to the grass. And we see that the seed mix better captures phosphorus compared to bare ground, which can make applying a diverse species mixture within bare ground soil a viable management practice. So here are some of the friends that we would see within the shade house throughout the course of the experiment. There's some fun frogs, maybe a mole, not too sure. And then we have our little caterpillar. So I just wanted to thank you all so much for coming out. Thank you to the Florida Department of Agriculture, National Cattlemen's Beef Association, and USDA LTAR, whose generous funding supported our ability to do this project. Thank you to Betsy for being so kind and supportive with this project all the way through. Thank you, Karen, for letting me tag on to your overseeding study and providing so much great advice for encouragement. Thank you so much to everyone who took the time to provide great insight and assistance throughout this project. Thank you to all the Archbold and Buck Island staff for allowing me to learn so much from you. And last but not least, thanks to all those who helped organize and set everything up with me today. I really appreciate it. All right, so any questions?
Okay. All right. Do we have any questions online? All right. Here's a question.
I wanted to know a bit more about the normalization, normal distribution test you did. Like, why were you looking for normal distribution, like in the result in the first place?
So I believe that we look for normal distribution just because it helps us really just analyze the data maybe in a better way. Yeah, it helps us meet the assumptions. So that's why we try to fit it if it isn't normal, so we could come to a more like significant conclusion about that. Yeah.
Yes. So because this experiment had the categories that we were trying to analyze, it would be the analysis of variance, or ANOVA, that we would use if it was a normal distribution. That means the data would have a sort of bell-shaped curve, but all of our data was skewed. So because of that, it didn't meet the assumptions of the ANOVA, and we had to use the non-parametric Kruskal-Wallis test.
I want to know why specifically you use daikon radish as your taproot species, like in particular.
Yeah, sorry, we have to move this microphone around.
So we use this as our taproot species because this is just one of the taproot species that was within our winter seed mix. Were there other taproot species? Yeah. Yeah. So that's why we ended up using the radish. And so I use these species because it was part of the overarching overseeding experiment that's going on the ranch. So that's kind of why we chose the individual species.
Yes. And another thing, we worked with a person from a seed company, and he recommended daikon radish and turnip for our area. It's really hot in Florida, and these species typically grow in cooler weather. So that's why we call it the winter mix. Like, we would plant these when Florida has cooler weather in over November through January. So they were supposed to be best adapted for these conditions in Florida in the dry season.
Thanks, great presentation. I think your phosphorus data I was particularly interested in, because I think we normally think of functional groups, right? You mentioned using 2 legumes and 2 grass species, but those were the most different from each other. And so, I mean, you suggested that vetch and clover maybe have some differences, but I'm curious if you think for this area, if the idea of functional groups is just not going to work. Does every legume or every grass species need to be tested? Or do you think there are some specific things that people should look at to help figure out what species within a functional group might be better or worse preventing phosphorus leaching?
Yeah, so I think that that functional groups probably aren't very representative at predicting anything. I think a lot of it comes down to more of the species differences. So we probably would have to look more at like each individual species to choose which one would be better. I think it would also be interesting to see which species for which seasons are the best, because I assume that that is also probably different.
Karen, do you have any thoughts about that question? Just the different root structures, which you said a lot.
Yeah, so we did measure a lot. All these pots we then harvested at the end. We took out every single individual plant's roots and we washed them and we looked at the specific root length and the biomass. We looked at rhizomes versus like the taproot versus the roots. And we looked at above ground biomass as well. And we tried to relate those different factors to these different nutrient concentrations. And we really did not see a strong significant relationship between them. I know Bahia grass was just so large and weighty that I think when we took it out, we did see that when there were more fine roots, we did see I think we did see an ability to capture more leachate. Now I can't remember, but that is something we do hope to explore later on. It does seem like each particular plant just does well individually, even when it comes to establishment. Like in the field, the first year we saw turnips were barely present, but daikon radish did really well. And we barely see vetch, but then we see clover establish well. So I do think they are very individual in their ability to establish and capture nutrients.
And just a quick follow-up, did both of the legumes have nodules? They both noduled, okay. Thank you.
Was my question.
Sorry.
Did they with the nodules like did that, I don't know how much data you took on it, but could that explain like maybe the vetch wasn't nodulating quite as much?
So I'll answer your question for you. When we scan these roots for specific root length, we do have scanned images of them. It could be possible we could isolate the nodules and look. We did see them. It was pretty cool.
Martia, our phosphorus expert.
Yeah, I was just struck by the sheer variability between the different species, both in terms of phosphorus in leachate and nitrogen forms in leachate, right? And I was thinking it could be useful to also look at the nitrogen and phosphorus in plant tissue, because different species have different entropy ratios, and that trying that with roots could also help understand maybe like why are like understand the different species differences in leachate quality. Yeah.
So I think it would be interesting to look at what's coming down through the atmospheric, like those forms of these concentrations, how that the concentrations that are within the plants, the concentrations that are within the soil, and the concentrations that are within the leachate, just to see better how the nutrient cycle over time. All right, any other questions? Or
All right, let's give a hand for Katie. Thank you so much to everyone tuning in. And I think our next seminar is on August 1st or something like that. So hope to see you here again then.